Dios, me asustó.
Okay, can somebody confirm you can hear me?
Can hear you.
I can
Thank
hear
you.
you.
Let me share my screen. My computer's being a bit slow because it's still working on the lecture video, so please bear with me. There we go. All right, before we begin, are there any questions? Today is Thursday, September 25th, and we are supposed to be talking about Lab 01, the ubiquity of microorganisms. Remember, the worksheet to this lab, as well as Lab 00, must be turned in by 8 a.m. Friday. Otherwise, you will be dropped from the class. The environmental quiz is due, I think it's due this Saturday. And then the online plagiarism quiz is due this Saturday at 11:59 p.m. On the plagiarism quiz, you must achieve 100% on the quiz. Oh, good. The lecture file just finished. That's what that was. You must get 100% on the online plagiarism quiz, or you will get 0 on the infectious disease project. In all my years, I've only had one student who got 0 on the infectious disease project or didn't get 100% on the plagiarism quiz. So please take the plagiarism quiz. And if you do not get 100%, you just need to take it again. I do not recommend that you guess, because if you just guess, and then you take it again and you guess, you get something wrong, and you must get 100% on the plagiarism quiz. If you just guess without learning the material, you may not get 100% on the plagiarism quiz. You should also read the infectious disease research project file called the project instructions in Canvas. I'll have to make an announcement of this. I forgot to mention that to my 230 lab, but let me show you where that is. There it is, week 2 lab, the infectious disease research project. I'll be talking about this next week, and you should at least look over the project before I begin talking about it. Any questions about any of that? Make a note of that. It's funny, I'm always making mistakes in the 230 lab. All right, any questions about anything? If not, let's go on to the lab. Lab module 01, ubiquity of microorganisms. With each lab, there will be some reading associated with the lab. For this lab, you should read page 157 to 162 of the custom edition of the textbook for Clark, meaning your textbook, on culture media. You should also read a page 162 to 163 on how obtaining pure cultures, how that was done. There's some video clips in today's lab. actually show one because it's so short. The learning objectives for this lab, one, know that microorganisms are almost everywhere on the planet Earth. And two, describe the purpose of culture media. Three, describe what colony abundance and colony morphology are. And then four, define the following terms. What's inoculation? Ubiquity of microorganism. What is something that's sterile? What is isolation? What is incubation? What is in vitro, like when we put the plates in vitro culture? What is a culture media? We have different types of culture media, a broth, an auger, a slant, and a deep, and I'll talk about those in this lab. What is a fastidious organism? Like Streptococcus pyogenes is a fastidious organism, and it cannot grow on, well, most media. It has to grow on a specific media, and I'll talk a little bit about that. And then what's non-selective media? What does ubiquitous mean? And what is a colony? Any questions about any of these objectives? All right. Microorganisms are organisms that are too small to be seen with the naked eye. This does include viruses and prions, which is in this group, even though they aren't really an organism and they're not really alive, at least in the traditional sense. And, well, they don't have cells. We'll also include helminths, worms in this group in our studies, even though the adults are not microscopic. As I mentioned, the parasitic beef tapeworm is something can grow up to something like 30 meters long, so it's not microscopic. However, the infective stages of the worms are microscopic, as well as some of the other life stages of the worms. The phrase ubiquity of microorganism refers to the concept that microorganisms are essentially everywhere on the planet Earth. Something that is sterile means that it's free of living cells. So if you were to go into the microbiology lab at Clark and I were to hand you sterile media, that's what I mean. It's media containing no living cells. And how we sterilize things is you either treat it with a chemical or with radiation, or more likely in the microbiology lab, it's been put in the autoclave to sterilize the media. In this lab, we're going to investigate the abundance and diversity of microorganisms in common environments. We'll also learn some basic lab protocols. All right, any questions so far? A culture media, also called a growth medium, although I never call it a growth medium, but is a culture or grow culture medium, is a sterile laboratory preparation that contains a variety of nutrients required for growing many microorganisms in vitro. In vitro meaning in a lab. Culture media may be liquid. These are called broths, and they're commonly used in a test tube because if you put a liquid in a petri dish, it's just too easy to spill, make a mess. Let me show you down here. So there's a broth, a liquid culture media. We also have solid culture media, and we usually put agar in to solidify the media, but you could use another ingredient such as gelatin or starch to solidify the media, but most of the time we use agar. Agar can be put into a test tube, in which case we call it a deep. So this right here is what a deep looks like, just a tube of media, solid, that we allow to form a solid, meaning the agar cools, and when it's a liquid, it's hot, and we allow it to cool, and it just forms straight across. So that's called a deep. And we also have slants where we put the liquid agar in the tube, and then we hold the tube at an angle, allowing the agar to cool, creating the slant. And this is called a slant. We can also grow or put agar into a petri dish. When it's hot, pour it in, and then allow it to cool, and it'll form a solid surface, which you can see that there. That one it's not so easy to see, but that one you can see, probably because it's cloudy or translucent or opaque, one of the two, whereas that one looks transparent. But there is agar in a petri dish. Any question about any of that? We have a video here for you to watch on preparing pour plates. Note how the culture medium needs to be sterilized prior to its use. An important question to consider is why must a culture medium be sterile prior to it being used? Oops, lost my place there. Let's move down. Boy, that's pretty bad. I'm talking to myself.
So that it doesn't contaminate the specimen.
Yeah, you need sterile media prior to its use, because if it isn't sterile and you pour it into the plate, you'll have all sorts of organisms growing on the plate besides the one you streak on the plate. There are a variety of agar media that microbiologists can use. We'll talk about that later in the term. Today, we're only going to talk about general purpose media. This is a media which is non-selective in function. It's usually of complex composition, meaning it's composed of many different molecules. And the general purpose media support the growth of most non-fastidious organisms, like E. coli or Staphylococcus aureus. A general purpose media cannot support the growth of a fastidious organism, such as Streptococcus pyogenes. Streptococcus pyogenes, if you do not know, is the organism which causes strep throat. And it will not grow on a general purpose media because it requires certain additives to be added to the media before Streptococcus pyogenes can grow. So a fastidious organism is one that's picky, and you need to add certain ingredients to the media before it will grow. Any questions about any of that? All right, let's return to a general purpose media. There are three very common general purpose media in microbiology, and we'll use these three throughout the term. Let's talk about the third one first, nutrient agar. It's the simplest. It's also the oldest, I think. All it has is three ingredients: ground up beef extract, sodium chloride, water, and maybe one other ingredient. I don't remember. And then you mix it up and you grow things in it. So very simple nutrient agar. Tryptic soy agar is similar, except it has, instead of beef extract, it has partially digested protein extracts from protein isolated from soybean. Why it's called tryptic soy agar is because we get this protein, partially digested protein, from soybeans. Tryptic soy agar. And a nutrient agar, the abbreviation is Na. Tryptic soy agar, the abbreviation is TSA. And then we have nutrient agar with yeast extract. It's a little richer than nutrient agar. It's the same, except we add yeast extract to it. This is just ground up dry yeast. And this one we call Na. All of these are general purpose media. They can grow many organisms, including E. coli, Staphylococcus aureus, Staphylococcus epidermidis. They can grow most organisms except for a fastidious organism, such as Streptococcus pyogenes. Any question about any of that? Okay, in the lab, you need to appropriately label all your plates and all your tubes correctly. And you do that prior to using the tube. There are 5 items that should be or on the label of the plate. You should put your name or your initials, the date, the type of medium it is, like nay, the inoculum, that's what you're going to culture on the medium, and the incubation temperature, which this one is optional. All the others are required. The incubation temperature is optional. Here's an example. The person who made this plate was Travis. They're growing E. coli, so he wrote E. coli. There's the date there, 9/10. I can't read that date, but it might be 2001. And then the media, TSA, tryptic soy agar, and then grown at 37 degrees. Remember, everything is required except for the temperature, which is optional. You should put the right on the bottom of the plate around the edge so that this region of the plate, you can actually see the growth on it. And if you don't write on the bottom of the plate, you can write on the bottom edge of the plate, which I don't usually mention to my students. Hopefully you can see this here. Make it in front of my face there, where you write on this side edge of the plate. But if you don't do it on the side, then write on the bottom. Why do we write on the bottom? Because the bottom of the plate contains the media, which will contain the cells. And if ever the lid gets separated from the bottom, we have the information of what's in the plate on the bottom of the plate. And how important is this? I was once in a lab where a lady was taking a whole bunch of trays, at least 100, maybe as many as 300 trays on a, I don't know, tray that she was holding the plates on. And as she was walking, she tripped and she spilled all the plates on the floor. All of the lids came off the plates, and she had written all of the information about the plates on the lids. And so nobody knew which plate went to which lid. And so all the plates had to be discarded, and the experiment, which had hundreds of plates, had to be repeated. Any question about any of that? Now, if she had not tripped, everything would have been fine. And if she had written everything on the bottom of the plate, everything would have been fine. Any question about any of that? So always write on the bottom of the plate. Another reason for writing on the bottom of the plate, this is a minor reason, is when we put the plates in the incubator, you put them in the incubator upside down. Having the information on the bottom of the plate means you can easily read it and find out which plate is yours, but that's just a minor reason for doing it. The reason why we put the plates in the incubator upside down is because the plates have agar in them and there's moisture in the agar. The temperature of the incubator draws the moisture out of the agar and it will condense on the lid, bottom of the lid, forming a droplet. And when that droplet becomes big enough, the droplet will drop down onto the plates. That will disperse your colonies. So instead of getting individual colonies growing everywhere, you'll get a whole bunch of growth with all the colonies mixed together on the plate from the drop of water dropping onto the plate. So that's why we put the plates in the incubator upside down. Now, when water comes out of the agar, it will condense on in this case, the top of the agar or the bottom of the agar, depending how you're looking at it. The droplet will form on the agar, and actually the agar will wick the droplet so it will not stay in one location, that it'll be wick throughout the auger, and that won't form a droplet and it won't drop down. But even if it were to form a droplet and drop down, we don't have any colonies on the lid. So it doesn't matter if the droplet were to drop down onto the lid. So that's why we incubate our plates in the incubator upside down to keep the droplets from forming on the lid and dropping on the colonies, spreading the colonies around, in which case you have mixed colonies, you don't have isolated colonies. It also reduces the risk of contamination. The droplet dropping from the lid down to the plate is probably going to be sterile because the majority of the lid is sterile. There is one region of the lid, though, that is not sterile, and that is this region right here, the edge of the plate. That is open to the outside, and it is not sterile. If the droplet of water were to move or form on the edge and then run down the edge, if the droplet were to get into the plate, such as when you're taking off the lid and you're moving it this way, for example, if it were to drop running on the edge into the plate, you would contaminate the plate. So also having the lid on the plate in its normal position, like on the top, and then sticking the plate in right side up, you do increase the risk of contamination. If you put it in upside down, you reduce the risk of contamination. Any question about the plates and how we grow them in the incubator upside down? All right. Inoculation is the placement or transfer of microbial specimens onto a sterile culture medium. Incubation is the placement of the inoculated culture media in an appropriate physical environment for a period of time to allow the microbes to grow. And here we put the plates in this contraption to incubate them. And this is actually called an incubator. This is a complex incubator that can control the humidity, the temperature, the time. The time is when you put it in, and then when you pull it out. It's up to you to determine the time. But this one is complex. It can also control the amount of oxygen in the incubator and the amount of CO2 in the incubator. In the microbiology lab at Clark, we only have simple incubators that do not control the amount of oxygen or CO2 in the incubator. It only controls the temperature, which you set the temperature at, and then you shut the door and the thing will heat if it's too cold. It doesn't refrigerate, but usually our temperature is above room temperature. And then you put water in this container here, and that makes the air humidified. The warmth of the incubator warms up and makes the air humidified. The humidity is important because if you put a plate in the incubator without any humidity in the air, the plate will dry up in a few days' time. If you, on the other hand, put water in the incubator, creating humidity in the incubator, the plate will survive for at least a week, maybe 2 weeks in the incubator before drying out. And once the plate dries out, the bacteria will stop growing if they don't die. Any question about any of that? Let's see, I think it was up here. All right, when we take a look at the plate, we see bumps on the surface of the auger. Each of those bumps is a colony. A colony has to have 1 million cells before it'll be big enough for you guys to see it. 1 million bacteria cells, I should say. Bacteria colonies tend to have a smoothly textured and a complete colony that differs from many fungi which have a not smooth surface, not smooth edge or regular edge. Like that's a bacterial colony. Let me blow this up a little bit. Get that out of the way. There's the edge. You can distinctly see that the auger is here, and then right there is the edge of the colony. This one here, it's hard to see where the colony end is and where the auger is because it's kind of hairy. It doesn't look very hairy there because it's blurred, but let's go down a little. There, you can see the hair's a little better there. And that's just the way fungi tend to grow. They're either furry or hairy. They're also kind of rough. And non-distinct colonies, the bacteria colony is usually much more distinct than a fungal colony. A colony is made up of 10 to hundreds of millions of cells. A colony must have at least 1 million cells before you can see the colony. Under 1 million, it'll be too small to see. Each colony may be a descendant of one cell that was placed there initially before you inoculated the plate in the incubator. And then that one cell will grow into two, two into four, four into 16, etc., until you get at least 1 million cells before you can see the colony. And bacteria tend to grow by binary fission, where one cell tends to split into two. Let's see if I can watch this video. Thought I clicked on it, but it didn't seem to take. There, we don't need that anymore.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about that? Let's see. All right, so I said in theory, one cell could lead to the formation of one colony. Yeah, you should understand that a colony can be formed by more than one cell. In fact, about 30% of the colonies on this plate will be formed by two or more cells. And by more cells, meaning a clump of cells. In fact, I have a feeling that that colony right there was formed by a clump of cells. And the reason is this colony is bigger than these colonies, which are bigger than these colonies. These colonies here are smaller because there's competition for resources. And so the colonies, these are the same species, by the way, except for that one. These colonies are close together, so they're competing for resources. So they're smaller than these colonies, which are separate and not competing for resources. That one is bigger than these two, so I'm pretty sure that one was formed by a clump of cells, and these two by either one or two cells. So, because they're smaller than this one, and yet they have more resources. This one is competing for resources with this colony here. And these are competing with resources for all of those colonies there. So that's why those are smaller than these two, for example. Anyways, any questions about any of that? So when we're looking at colonies on a plate, remember that about 30% of the colonies on the plate are formed by more than one cell. And if a colony is formed by two or more cells, the colony may not be pure. Now this looks like a pure colony, so maybe the clump of cells all derived from a single colony, a single cell, but it's definitely formed by more than one cell because that colony is much bigger than these colonies. And you don't need to know that. I'm just giving you an example of that being a colony that's too large for what it should be. It's probably because it was formed by a clump of cells. And you should know that 30% of the colonies on this plate are formed by more than one cell. All right. Colonies are not visible in a broth culture, and that's because in a liquid culture, all the cells mix together and you see no colonies. They just grow all together. And let me see if I can find a picture of that. I thought I had one in this lab, but I don't. Good images. Yeah, there's one there. Let's take a look at this one. There's several here. All right. Cells are growing there. That's why it's cloudy. Cells are growing there. It looks like this one, all the cells are growing only at the bottom. This one, all the cells are growing mainly in the media. And that's why it's very, very cloudy. You cannot see any colonies in any of these. You can't even see any colony in that one. There are often obvious visible colony morphological differences between different species of bacteria. And these give rise to what we call the colony characteristics. The colony can have a different shape. The colony can be circular, rhizoid, irregular, filamentous, or spindle-shaped. Most colonies are either circular or irregular, but you do have some rhizoid, filamentous, spindle. The colony can have an edge to the colony, like this colony has an entire edge, and that's just a smooth edge, entire. Or it could have an irregular edge, which would be undulate, lobed, or curled. I'll never ask you to tell me, is the margin of the colony undulate, lobed, or curled? Because it's too difficult for even me to distinguish between these three. But I may ask you, is the margin of the colony entire, or is it undulate, lobate, or curled? Any question about any of that? And I could also ask you, is the margin of the colony entire, or is it rhizoid or filamentous? I've never asked you, is the colony rhizoid, or is it filamentous? Because it's just too difficult to separate those two out. The colony also has an elevation. Usually to see the elevation, you have to look at the colony from the side. The colony can be flat, it can be raised, that's like a mesa, it can be convex, it can be super convex, which is called pulvinate, and it can be umbinate. I would never ask you, is the colony convex or pulvinate? Because it's just too difficult separating those two out. But I could ask you to separate the others. Here we're looking at a plate right here. Can anyone tell me, is this colony flat, a mesa, um, convex, or umbonate? Can anyone
Flat.
answer?
Flat.
Flat? Anyone have a different answer?
I would guess convex.
It's really hard to tell because we're not looking at this colony from the side. And when you're looking at the colony from the top, it's almost impossible to know. I think,
Could it be rise?
oh, I'm sorry,
Rise.
uh, you mean rhizoid? The elevation was what I was asking, and or raised, maybe you were saying. Uh, it it probably is raised or convex. Okay, it doesn't look umbonate, although you can't really tell that from looking on the top. And I don't know about separating those two. I'm not going to do it. Like I said, I can't tell. It's probably convex. It could be raised. I don't think it's flat. And the reason is I can see a little bit of difference between the center of the colony and the edge. Right there. So I don't think that one's flat. That one could be flat. That one could be flat. But that one I don't think is flat. And like I said, when you're looking at the colony from anything by the edge, it's really difficult to tell. And so I will never ask you to that question on a picture like this. And I probably won't ask you the picture because to take a picture of the colony from the side is really pretty difficult. You need to look at it, the colony from its side. There's a different size to a colony. If the colony is smaller than the tip of a pencil or pen, the colony size is punctiform. And punctiform means essentially a pinprick. If the colony is around the size of the tip of the pencil or the tip of a pen, the colony is small. If the colony is as large as or larger than the eraser or the other edge of the pen, then the colony is large. If the colony is in between the eraser side and the tip of the pencil or the tip of the pen, then the colony is moderate in size. Any question about any of that? You can tell is actually this is the size. I don't know if you can see that, but I'm touching it to the screen. You probably can't see that because I don't even know if you can see the tip of the pencil. But that's if it's that size or larger, it's large. And let's go here. Yeah, small is actually a little larger than that one. The colony can be smooth or rough. I'm not sure why, but students often have a trouble telling whether the colony is smooth or rough. Is that colony smooth or rough?
I think it's smooth.
Yeah, that's a smooth colony. And is it glistening or shiny, or is it dull? That one right
Shiny.
there. Yeah, that one's shiny or glistening. Those two we use the same, shiny or glistening. So that's the appearance. The colony can also be pigmented. If the colony has any color, it is pigmented. It's non-pigmented if the colony is white, off-white, tan, cream-colored. Maybe black too. I don't know about black because black's actually the absence of color. But normally bacteria, you don't see them black. So is that colony pigmented or non-pigmented? This one right here.
Pigmented.
Pigmented. How about that colony there? Pigmented or non-pigmented?
Non-pigmented.
Non-pigmented. Good. The colony can also have an optical property. Most colonies are opaque. Some colonies are translucent, usually a few on a plate. if you have a multiple types of colonies. A few will be translucent, and then very, very few will be transparent. A transparent colony looks like a drop of water on the plate. Very few bacteria are transparent, meaning very few bacteria species. In my entire life, I've only seen a few transparent colonies, and I've looked at a lot of colonies over the years. All right, so that colony right there is translucent. You can sort of see through the bacteria here. A little bit of this one is transparent, but I'm not translucent, sorry. But the center is definitely opaque. Most of these colonies are opaque, like that one is opaque, that one is opaque, that one is opaque, that one is opaque. Any question about the colony characteristics? It's just saying what some of them are. Like colony C, I say that's a convex colony. But when you're looking at it from the top, it's very hard to tell. Colony B is actually flat, and that also is very hard to tell when you're looking at the colony from the top. Colony abundance is simply the total number of colonies on an agar plate. Too many for me to count there, so I'm not going to use that one. But I will go right here. What is the colony abundance in this region of the plate here? So you don't have to do all of those, but just this region right here. What's the colony abundance? Come on. Somebody should guess.
12.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Yeah, that's what I got too. 12 is the colony abundance. What is the colony type or diversity in this region right here? Anyone have a guess?
2.
No. Yeah, I would say 2. That colony, and then all these others are probably the same species. That's a different species. So the diversity there is 2. There's just a whole lot of diversity there. I'm not going to even try because that's a different colony from that one. That's a different colony from that one. That's a different colony from this one. These two are different, so there's one. Well, that might be the same as there. Probably not, though. The edge looks different. So 1, 2, 3, 4. So just right there, there's 4. And there's a whole bunch of little ones. That's probably 5. Pigmented, 6. Another pigmented one, 7. That probably a different colony, although it could be the same as that one. There's another pigmented one. I don't know if I said that one or not. So there's another one. So there's a whole bunch of different types in there. Diversity is simply the number of different types of colonies. You use the different colony characteristics, especially color, shape, texture, etc., to identify different colony types. Realize with shape, you have to give a variety for the size. I mean the size. The shape probably can be used. Like that's clearly a different colony than that one. Not only is the color different, but the growth of colony, the protrusion of the edge there. And this is a round colony.
Quick question. So if they were the same color but different shape, that would count as 2, correct?
Say again?
Sorry. So if there was, like, let's say, for example, there was 2 on there, and then they were both the same color, but they were different shapes, that would count as the diversity of 2?
Yes, if you're saying
okay.
the colony is a different colony characteristics, which either the margin differs or the, where is it here? Shape differs. Like in a regular colony, even same color as a circular colony, those are different species.
Thank you.
All right. All right, here's an example. What is the abundance of wild cats in this picture?
7.
7 is correct. And what's the diversity of cat species in this picture?
2.
2 is correct. There's lions and there's 5 cheetahs. Somebody has their microphone.
Well, when you're looking at like the culture and you see some that are like so like different colors, are we and it asks like what is the colony diversity in there? Would you separate them based off of like colors too? Like they would be different in like if they were different colors, or are we going based off of like its shape or
You can use all of the characteristics to give the different species. Obviously, size, you have to allow a lot of range in the size, but if they have a different pigmentation, if they have different margin, you can say they're different species. Okay?
Yes, thank you.
All right. There's some terms you can learn for this lab. Somebody's hand. Don't need to look at the references. And then there's the answer to the question. So the abundance of colonies in the plate it asks in the question 80 to 90. Diversity at least 10. All right, let's go to the worksheet. You're not directly going to be answering or doing anything directly. I'll always provide pictures in the worksheet, and then you'll get your question and answers based on something like the picture or the information I give you. So in lab 1, we're asking the question about the ubiquity of microorganisms. We have a number of different environments sampled, and then grew the cells sampled in the different environments on general purpose media. For procedure 1, we're answering the question, what is in the air you breathe? There is microbes in the air, and we expose the plate to the air, meaning we leave the lid off for probably about 20 minutes, and then put the lid on, incubated the plate for 24 to 48 hours in a 30-degree incubator, and then we got these colonies growing from the air. So for procedure 1A, what is the colony abundance on this plate? Yes, I need to count all the colonies. What is the colony diversity on this plate? And you don't have to have an exact number, but your number should be close to my number. Meaning if you say something like there's 30 colonies and I get something like 29, that would obviously be correct. However, if you say the colony diversity is 30 and I say something like 3, then obviously that is incorrect. So you have to be close. Any question about colony abundance or colony diversity on this plate? So you don't have to have the exact number, but it has to be close.
So even those little tiny pinprick ones, are we still counting those?
Yeah, those right there are definitely colonies. So's that little one there. These right here is a good question. Oops, come on, didn't mean to do that. Let's go back there. These in there, I don't know what's forming those dots inside there, and I don't know if those are individual colonies or not. I can tell you that one right there, that one right there is a separate colony from that one right there. And you know, that looks like a separate colony right there from that one right there. So that one is a colony growing on top of a colony. That's a little one touching the colony there, and then that looks like a colony there, and there's a colony there. But these little pinpoint colonies, you should count them. Okay. And there's one there. That is the lid, so that's not a colony right there. Like I said, you only need to be close. Don't have to be exact. What's the colony diversity on this plate? You need to look at the different colony characteristics to get the number of species on the plate. Like if I were to look at these three colonies here, what is the colony diversity of these three?
3.
It's either 2 or 3. That could be the same colony, meaning same species. It could be different. I will agree that the size difference is different. And then the colony shape looks different too. But everything else looks similar between these two. The pigmentation is about the same. It is true the shape of the colony. This is, I don't know, unusual shape. And that's a circular one. So it's either 2 or 3 for those 3 right there. These are definitely different. And then procedure 1C. While you are inoculating the plate, what could you do to prevent contamination of the plate from airborne microbes? And you have to answer what you're doing while you are inoculating the plate. It cannot be something you do before you're inoculating the plate or something you do after you inoculate the plate. It's something you're doing while you are inoculating the plate that can reduce airborne contamination. And that's procedure 1C. You do have to think about this one. It is discussed in the lab, by the way. But that's not an easy answer, and students frequently get that one wrong. Probably because they're not thinking. They tell me something they do before or after they're inoculating. Procedure 2, what gets on your arms when you put your arms on the lab bench? What we did was we sampled the lab bench for bacteria and put it on the plate before. You can see all the growth on the plate before. And then we disinfected the lab bench and then sampled the lab bench and put it on the plate here. And this is what the after is. So the question is, did table disinfection work? It's either yes or no. And then the next question is, what is the purpose of allowing the table disinfectant to air dry? So what we do is, when we disinfect a table, we spray disinfectant on the table and then wipe it around with a single sheet of paper to smear it around. We do not grab a second piece of paper. Let me get a second one. And then wipe off all of the, I don't know if you can see this or not. Can you see my hand here? Getting a second piece of paper and then wipe dry the table, meaning we only wipe it one time with one paper, which is wet from the disinfectant, and then we allow the table to air dry. What is the purpose of allowing the table disinfectant to air dry? Or why do we air dry the disinfectant? Any questions about that? This is a thinking question. It's not that difficult once you think about it and get your answer. I think you can also do this by putting the question in AI and probably getting an answer, but I haven't done it. And you could get the wrong answer if you use AI. So you need to think about this one. I don't think that's stated in the lab there, but it's common sense when you think about it.
So that you don't contaminate the or like recontaminating by wiping it down?
No, that's not really why we're allowing it to air dry. And
It would be
I'm
less
not
of
going to
a,
give you an answer to this. You guys are supposed to answer it yourself. You can answer a question, but don't give me the answer. Okay? So is your question about procedure 2B a question, or are you asking for the answer? There was somebody else who was talking. All right. I guess they were asking for the answer. Like I said, I'm not going to give you the answer there, but just think about it. All right, procedure 3. What is on your hands? What we did was we sampled our hands before we washed our hands and then after we washed our hands. And when you wash your hands, you have to dry your hands so that your hands are comparatively the same dryness before you wash your hands and then touch the plate, and then after you wash your hands and touch the plate. When I was first teaching microbiology and doing an experiment like this, every one of my students was getting more growth from their washed hands than they were getting from their unwashed hands. And then I asked them what they were doing, because this is very unusual. And it turned out what they were doing is washing their hands and not drying their hands and then directly touching the plate. And the trouble is, wet hands wick the bacteria from your fingers to the plate. And so you have to have your hands relatively the same dryness from the unwashed hands to the washed hands. So you did have to wash your hands and then dry your hands using paper towels. And the students initially were refusing to dry their hands because they didn't want to put bacteria from the paper towels onto their hands. And let me tell you, there's very few bacteria in the paper towels, assuming they're clean paper towels. Any question about any of that? So you wash your hands or unwash your hands, touch the plate, and then you go wash your hands, dry your hands with paper towels, and then touch the plate. And then we see this amount of bacteria here and that amount of bacteria from the unwashed hand. The first question is, does hand washing work? And then the second question is, suggest one reason why hand washing did not completely remove the microbes from your hand. It may have to do with the normal microbiota of your skin and the strength of the soap and water to sterilize your hands. For example, if I were to wash my hands with chlorine bleach and then dry my hands, you would see much fewer bacteria than when we wash our hands with soap and water. And why might that be? Anyways, that's 2 hints for number 2. Students frequently get this one wrong because it's a thinking question and they just don't think it through. But if you think it through, you'll get it. And if you get your answer online, please provide a citation for your information source where you got your answer, including if you got your answer from AI. Procedure 4. When you kiss your significant other, what exactly are you giving him or her? What's on their lips? What's on their tongue? What we did was we had somebody touch their lips, the bottom lip, the top lip, to a plate, stick it in the incubator for 30 degrees for 48 to 72 hours, and then touch their tongue on the plate, and then stick it in the incubator for 48 to 72 hours. And this incubator is at 37 degrees. You should take a note that I've added a note above the picture talking about the tongue plate. So you should read that note and then answer for A, which plate has higher colony abundance, the lip plate or the tongue plate? And then for B, why do you think that this is the case, that one plate has higher colony abundance than the other? Even though you might expect the abundance of both body sites to be the same, they're right next to each other. You probably frequently or occasionally lick your lips with your tongue. So what might cause the observed difference in colony abundance? And you must, to get full credit on this one, give me 2 correct answers. Because there's at least 3. Students have actually come up with 5 reasons why one, the lip or the tongue, may have more bacteria than the other. Okay, so you
Wait,
must
I
give me
had
at
a
least
question.
2 reasons. Yes.
So since they incubated it at different temperatures, could that be the cause of the difference in abundance?
But they were incubated at different temperatures.
Yeah, because didn't it say 30 degrees and 37 degrees?
Yeah, they are incubated at a different temperature, but that's not the reason why they are incubated at a different temperature. Because if you were to grow them at the same temperature, you
Mhm.
would see very similar results to what were shown here. So
Okay.
that doesn't give rise to the difference in the abundance.
Okay, thank you.
Okay. But
I have
there
a
is
question.
C, it's asking the question, why was the tongue plate incubated at a higher temperature than the lip plate? So you do have to answer that in C. And this one is pretty obvious once you think about it. Why are we growing the tongue plate at a higher temperature than the lip plate? If you need to, you can take a thermometer to your tongue and a thermometer to your lips and answer the question that way if you want. But that really doesn't give rise to the abundance, the different temperature that we incubated at. Um, C is the reason why we're incubating at different temperatures.
I have a question.
Uh-huh.
Around the outer perimeter of the lips, all those little specks, do those count as colonies? Because they're kind of hard to count.
You don't need to give an answer here that the lips have so many numbers to count, meaning I'm not looking for a count of the numbers. Let me blow it way up. So the abundance is all of the colonies. I just need to know, is there more abundance in the lips or more abundance in the tongue?
Okay, so those
Yeah,
little
you should count these little things. I mean, you're not going to count them, but you just need to tell me. That's obviously one there. That's one there. That's one there. Okay.
Okay, thank you.
All right.
I have one more question on that too.
Okay.
So, since the tongue has a big portion of like the blurry colony underneath the multiple different ones, are those a bunch of little colonies that are just kind of globbed together, or is that one huge one?
You should read my note above the picture.
Oh, I see. Okay.
All right. Any other questions about procedure 4? If not, let's move on to procedure 5. Our microorganisms in the soil. We have a petri dish, and we took, I think, a gram of soil. Let me take a look here. It's not stated in there, but you take a gram of soil, put it in 9 mils of water, mix it up, and then you take the swab of the diluted soil, meaning it's diluted in water, and then you swab the plate here, and then you incubate the plate for 24 to 48 hours at 30 degrees C, and then we got this plate here. And procedure 5A, what's the colony abundance? Is it even possible to accurately answer this question? Please explain your answer. In your answer here, you could give me an exact count number or an approximate count number, or you could state something like It's not possibly to accurately give an answer to this question because of, and you have to give a reason because of, but you just need to explain your answer. Okay? Any question about procedure 5A? So what's the colony abundance on this plate? And then for 5B, is there greater abundance of colonies from the soil compared to the lip plates? So here we have the abundance on the lip plate, and there you have the abundance on the soil plate. Some students in the past have stated, well, there's many small colonies on the lip plates, so I think the abundance is more on the lip plates than the soil plate. But if you look closely at the soil plate, there are some large colonies, like right here, right there, right there, right there, right there, right there, and right there. There's a large number of large colonies, but there's a large number of little colonies all right there and right there, and a whole bunch of little colonies right here with some large colonies, like there's a large colony right there, but a whole bunch of little colonies. So there are a number of little colonies in the soil plate. You just have to look for them because, well, they're kind of hidden by the large colonies and all the colonies growing together. Like this region right there is mostly little colonies. So there are little colonies on the soil plate. And there are a number of big colonies here. There are a few big colonies on the lip plate as well, like that one there and that one there. So is there greater abundance of colonies from the soil compared to the lip plates? And that's your answer there. Any questions about this lab? All right, I'll be here until at least 8 o'clock to answer questions, help you with the lab. I'll go ahead and get started on the lab. Do remember, I will log off once the last student has logged off. So if you have a question, make sure you ask before the last student logs off.
Quick
I did
question.
have a
Oh,
question.
Go ahead.
So I know at the beginning of like the lab module, the one we were reading at first, it says to pre-read the tutorial and the page numbers. So I haven't received my book just yet, and it says it's coming. But is there any way we can get those pages before?
Yeah, in the last lecture, I told you I have the first 3 chapters of the textbook, links to it online. And then I have the chapter summaries to all the chapters online. Now, if that is the page number you need, you're great. Otherwise, you're going to be out of luck. You'd have to go look at the course reserve library to get the textbook and look at the page numbers. But I might have the page numbers you're requesting in the links for the first 3 chapters.
Yeah, I tried to check and I saw it's between chapters 5 and 6, but there's no pages for those. That's why I
Yeah, then you're out of luck.
tried looking.
You need to go to
Okay, so just wait until it comes.
Yeah, or you could wait.
Oh,
The trouble is you
all right.
can't wait. You got to turn in this lab by 8 a.m. Friday. So
Which is
do the
tomorrow.
best. Yep. Just do your best. Which I guess the 8 a.m. library Friday, the library is probably not going to open until 8 a.m. if even that. Just do your best.
Hey, I have
Somebody
the
has the
textbook and I
textbook.
can
Yeah, can you
email it to that
email
discussion
the person?
group.
The trouble is, can you scan it or take a picture of it?
Yes, I can
Great.
scan it and put it in the discussion board that we have open.
Okay, that'll work too.
Okay. It
Thank you.
just might be like 30 minutes or so.
Okay, I understand. Hmm. So you want to do it in the Zoom workbook?
No, the one where you said bring all of your questions and
Okay.
discussions
So
here.
you're talking about the student discussions. Yes, go ahead and put it
Yes.
there. That'll be good.
Okay.
Yeah.
And then I did also have a question. In the lecture, I was the one who had asked if we would have access to the slides. I was referring to the slides that you were presenting to us.
After I'm done with the lesson, I will make the slides available to you.
Okay. So they will be online tonight. Yes?
No, we're not finished with chapter one. So you won't get them until at least next Tuesday.
But up until the point that you finished, I guess today, that will be on YouTube, right? Like we could be able to
Oh
rewatch.
yeah, obviously the course will be on Zoom, so if I record it, it will go up on YouTube. So you
Okay,
can
thank you.
see it on YouTube. Yes.
Okay. I have a question.
Uh-huh.
The environmental respondus lockdown browser
Uh-huh.
is still locked until tomorrow, right? The 26th, yeah. And it says it's due tomorrow at 8 a.m.
That shouldn't be. Let me take a look at this.
Well, it doesn't say it's due at 8 a.m. on Canvas, but it does on your syllabus.
Oh yeah, the syllabus might be wrong about that. I have the quiz, environmental quiz. It's due on September 27th, and it opens at 12:01 a.m. on September 26th, and it's due by September 27 at 11:59. So I actually have 2 days to do that quiz.
Okay, thank you.
Wait, so it unlocks tonight at midnight and then it's due on the 27th.
Correct. Meaning you must submit it on the 27th. Let me go ahead and change that on the syllabus. I mentioned that when I said it. That one is not due at 8 a.m. Friday. It's due, um, I don't have enough room here, but I'm just going to say Saturday.
Are you able to unlock it a little bit earlier so we could do today, or does it have to be unlocked tonight?
Well, the quizzes are normally locked for all periods except for 24 hours. This one I've allowed you to have for 48 hours, so I thought that was an exception. You're wanting more than 48 hours, and I'm going to say no, because you have to get used to the fact that you have to take the quiz within 24 hours. Does that make sense?
Yeah, thank
Mhm.
you.
But you can get it done af after midnight tonight. All right, let me change that because that was a mistake. I don't know if you can even see this anymore. I'm probably not showing it. Nope, doesn't matter. I just changed the syllabus there, but I need to upload that new syllabus.
On the lab, are we able where it says like label where we have to label these specimens? Can we put it on the side or do you want us to put like a text box inside of the dish where it says to label?
I don't remember a question where you're actually labeling a petri dish. Is that one of the questions?
Well, where it says procedure four, it says label two nape plates. Oh, okay, I guess it's already labeled. Let me see.
Okay. All right, so I think you got it solved. Like I said, I didn't think you had to label a plate. Don't open anymore. It's too bad.
Okay, so just to clarify, we don't have to label a plate?
I don't remember a question where you had to label a plate. If there is a question, let me know and I'll take a look, but I don't remember a question where you had to label a plate for an answer for one of the questions in the lab. I'm sorry, I can't look at the moment because I'm actually doing something else for the course, but I will look as soon as I'm done here.
So Dr. Volp, if we don't do, say we don't do too good on this lab, which hopefully is not the case, do we have an opportunity to go back and correct or no?
In the lab, yeah, you don't get a chance to regrade labs this week. So lab 00 and lab 01 will be graded one time. Lab 2 next week will be graded 2 times. Lab 3 through 9 will be dependent on what the students vote for.
Okay, perfect. Thank you so much.
Okay.
You have a good night, Dr. Volp.
All right, you too.
Sorry, is it just me? I was just working on the lab. But
It's fine. I don't know. You're the only one here. Let me take a look. I'm not there. I'm working on the course and another thing. No, there's 2 people here. Of course, I
oh,
won't be
okay.
the other person. Yep, Jennifer, you're the only one, but that's fine.
All right. Well, I'll log off then. I was just finishing it up, but thank you so much. All
You're welcome.
right, bye.
Bye. Whoops.